What is epigenetics? You're an excellent showcase of epigenetics. You once started out as a tiny fertilized cell called the zygote, and then grew to become a mass of 70 trillion cells. With the tiny exception of a tiny amount of cells in our immune system, all those cells are exactly the same at their genetic level. Yet, our cells in our bladder are so vastly different in their structure and function than the cells in, say, our eyeballs. If we express only the liver genes out of the DNA, we're going to create a liver cell. Now, again, all our cells have the same DNA, but they're not expressing the same genes at the same time. So how do we control which genes we're going to express? This is where epigenetics comes in. The word epi means at, on, or as well as. This means there's something else operating other than the genetic code. Here's a formal definition for you. Epigenetics is a process in which stably heritable phenotype results from changes in a chromosome without changes to unique DNA sequence or structure. Let's break this down a little. Remember, phenotype is our physical characteristics or appearances that result from genotype. And if something's heritable, this means it can be passed on to offspring. Epigenetic modifications are essentially changes in phenotype without changes in genotype. There are three mechanisms of epigenetics that cause epigenetic modifications: histone acetylation, DNA methylation, and microRNA. Now, you've seen DNA represented in the classic double helix twisted ladder structure, but it doesn't exist in our cells as a long string of molecules. Histones are proteins around which DNA is tightly wound. This is how we manage to compress and compact miles of DNA into the chromosomes that we find in every nuclei of every cell in our body. Now, in order for a gene to be expressed, its DNA has to be unwrapped from a histone so it can be transcribed, and thus the gene can be expressed. In order to get a histone to unwrap its DNA, it must first be acetylated. For our purposes, our DNA will be represented by this wire, and our histone will be this Play-Doh clay cylinder. These toothpicks will act as the tails that will stick out of our histones to receive chemical signals. This colored tape will mark the gene we're going to focus on. The DNA will be at various states of being coiled around the histone. Where's the gene we marked on our DNA? It's become so tightly wound around this histone that the DNA that makes the gene can't be read and transcribed, so the gene can't be expressed. So how do we get the gene to express? A bunch of chemical tags called acetyl groups will attach to the histone tails. These acetyl groups will decrease the attraction between the histone and the DNA it's so tightly wrapped around, urging this histone to let go of some of the DNA it had wrapped around it. And look what we have here, our gene. The DNA in this colored section of our gene can now be read and transcribed, thus allowing our gene to be expressed. In DNA methylation, we add a methyl group to the cytosine bases in a specific gene. Now remember, we have 4 different nucleotides: adenine, thymine, guanine, and cytosine, the rungs inside the twisted ladder of DNA. When we methylate cytosine, we're adding a methyl group to a specific rung in the ladder. It's like turning a switch off. This is the gene, and this is its promoter. If the cytosine bases are methylated in the promoter, DNA polymerase cannot bind to the DNA and read it. Thus, the rest of the gene is left unexpressed. We're turning that gene off. We can't make RNA, and we can't make those proteins in with the methyl groups blocking the code from being read. DNA methylation suppresses gene expression. Back to the idea of the differentiation of your cells from the initial fertilized embryo. If we want a heart cell, we methylate all of the genes that don't make that heart cell. The same thing is going to happen for all the other cells that form our body. The third mechanism of epigenetic change is through microRNAs. This gene is transcribed into RNA. This is called messenger RNA. There are microRNAs which do not code for any protein. Their only function is to sabotage the messenger RNAs, thus halting the synthesis of the protein. Even beyond the level of DNA, epigenetic mechanisms, including microRNAs, can control gene expression. It's essential to understand that your epigenetic modifications don't change your DNA sequence, but rather the way your DNA is transcribed and thus the way your genes are expressed. Your genome is your hardware, and your epigenome is a software that defines how that hardware is used. Now, your DNA is fixed for life, that's a given, but your epigenome is truly flexible. Your epigenome is what defines your genomic landscape. It's what changes it in response to your rapidly changing environment. Here's what we know. Epigenetic information can pass on from parent to offspring. Epigenetic information can also be influenced by our environment. But this duality leads us to another question. Can parents pass on their environmental responses to their offspring? We see this in an experiment where researchers exposed mice to the smell of cherry blossoms and subsequently gave them a mild electric shock. Now, of course, after several rounds of this treatment, the mice exhibited very nervous behavior, visibly shaking when they picked up on the smell of cherry blossoms. Now, the really fascinating thing that occurred in this experiment is that the offspring of these mice exhibited the same nervous behavior and visible shaking when they picked up on the smell of cherry blossoms too. These mice never went through the same shock treatment of their parents, yet picked up on the same behavior. Therefore, this puts a lot of evidence towards the idea of parents passing on their behavior to their offspring. Epigenetics and its implications are everywhere. It's still a rapidly expanding field of science. In some diseases like cancer and diabetes, enzymes that regulate epigenetic modifications malfunction, thus creating too much or too little of a protein, and this helps with disease progression. And from what we understand right now, simple things like what we eat, if we smoke, or the levels of stress in our environment can affect the way the genes are expressed. And this can even be passed on to our future generations. A genetic trait is not just a product of genes, it's also a product of an environment. Any person's genome is decided upon by any number of decisions that their ancestors may have taken. Right now, you're making decisions that are going to be affecting people who will be alive long after you're dead. No pressure. Thanks for watching.